In this key area, 3.2, we're going to be looking at plant and animal breeding and manipulation of heredity. So first of all, we're going to give some examples of characteristics that have been selected for by breeders and explain the benefits of that. Now, for hundreds of thousands of years, breeders of crops and livestock have been manipulating heredity. In other words, they've been doing selective breeding. They've been choosing animals that have got desirable characteristics and then trying to selectively breed them to enhance those characteristics. And examples of these we can have a wee look at are, well, this isn't actually really looking at food because we don't eat dogs, well, certainly not in this country, but selective breeding has been taking place with dogs for hundreds and hundreds of years, where certain characteristics are selected for depending on what it is that you want your dog to look like. So all dogs are originally descended from wild dogs, but over time what has happened is the certain characteristics have been selected. Here we've got carrots here that have been selected for, in this case here, it's some of the different colours that are there. I don't know if it's anything to do with taste as well. Down here there's some examples of maize. So here we would have had, say, a wild strain maize, and then over time it's been selectively bred to produce a much larger corn cob with larger actual kernels on it as well, and thus making it more desirable to grow. And then over here we're looking at the ancestor of brassicas here, and what has happened is that over time breeders have selected for various different parts of the leaf. So for example, you've got here selection for terminal buds, so you end up with your cabbage. You've got selection for the lateral buds, so you end up with the Brussels sprout situation. You've got selection for certain leaves, so if it's a plant that's got larger leaves, it's been chosen and selected over time. And you've got your kale, or you've got selection for flower clusters and you end up with your cauliflower, or stems and flowers, you end up with your broccoli. So all of these are ways that breeders of crops or of animals have been choosing certain desirable characteristics. Another example here is looking at the super cow. This picture that's down here, you're looking at this very, very muscular cow. Well, it's a bull in this situation here. These are called Belgian Blues, and they've been selectively bred to have this large muscular physique in order to be producing more meat. If you have a look at the top here, you're looking at a wild cow, what it would look like in the wild, and you'll see that over time that has been selectively bred to produce a beef cow, which has got more meat on it, and you can see the differences that are in the diagram. But when we're looking down here at this Belgian Blue, it's been even more selectively bred. It's got a certain genetic composition that results in these large pieces of muscle being produced, and these obviously make very good large cuts of meat for the farmer. There's a bit of controversy over the breeding of these super cows, but the bottom line is that selective breeding is done in order to produce a new and an improved cultivar of a plant or a new or improved breed of an animal that will provide a sustainable source of food for humans, because that's what we're talking about in the long run, producing more food. So here's some examples of hereditable characteristics that you might want to be selectively breeding for. So you might be looking for a higher yield in your crop, so in the increased mass of food by a wheat crop, for example, or some other crop. Or you might want to have a higher nutritional value in it, so you might want more protein, for example, in a soya crop, or you maybe want more vitamins in it. An example of that what we come on to look at is looking at rice that has had more vitamins added to it, albeit that's been done by genetic engineering, not by selective breeding. You may be looking for a crop that has got some resistance to a specific pest. So example of that would be eelworms in tomatoes, or resistance to some sort of disease, and an example of that is potato blight, and we'll have a look at that when we go on to do our game called Saving Our Spuds, and it's looking at this problem of potato blight. It might be that you want to have some sort of useful physical characteristic there. So you might want to produce a crop that's got some kind of uniform height, not too tall, not too short, but just perfect for mechanical harvesting. Or you may be wanting to select for a crop that can thrive in a particular environment. An example of that would be maybe the ability of maize to grow in a cold and a damp climate, in other words, in Scotland. Now we're going to go on and have a look at plant field trials. So maybe you have selectively bred a plant and you think that it's better in certain situations. Or you've maybe done that by selective breeding or by genetic breeding, but you want to compare the performance of two different plants in a similar set of environmental conditions, and then compare them, the effect of them in different environmental conditions. So, as I've said, a plant field trial is a type of investigation. It's set up to compare the performance of two different plant cultivars. By the word cultivars, we just mean the plant crop. So you might, for example, have produced you've got a genetically modified crop, and you want to compare that to the non-genetically modified. So you want to compare them in the same set of experimental conditions, or you want to find out the effect of different environmental conditions on the new cultivar of the crop plant. So let's imagine we're setting up a field trial and really looking at this situation here where we're not comparing two different plant cultivars, but we're comparing a new plant that we've got, or a new cultivar of a cereal plant, for example, and we want to find out, or we want to investigate the effect of the concentration of nitrogenous fertiliser on that new plant. So if you're a farmer and you've got a new cultivar or a new species of cereal plant, or a new crop that you've not grown before, you might want to find out what would be the best level of fertiliser that was on it. Or if you're somebody that is selling that crop seed to farmers, then you might want to provide them with that information of how much fertiliser should be given to the plant. Does it use more or does it use less? So you would set up a field trial. So once you've investigated, or once you've decided what you're going to investigate, there's these things that you need to consider: the selection of the treatments you're going to be used, the number of replicates to be included, and randomisation of treatments. So we'll look at these individually. So selection of treatments and replication. Now, what happens is you've got an area of land, and this is our area of land here, this box of squares that is here. And it's divided into equal-sized portions called plots. So each one of these is a plot. A field trial involves treatments, and a treatment refers to the way in which one plot is treated compared with other plots. So for example, one plot might be given a high concentration of fertiliser, so we'll give that A for a high. Another plot might be given a slightly lower concentration of fertiliser, another plot lower yet, and another plot even lower yet. So you've got your crop growing in each of these plots, but you're treating each of these plot areas differently with different fertilisers. In other words, you've got 4 different treatments that are going on here. It doesn't need to be 4, it could be, you could just have chosen 2 different treatments: high concentration of fertiliser, low concentration of fertiliser. So the only variable you're changing here is the concentration of fertiliser, and everything else has to be kept as constant as you possibly can make it to ensure a valid comparison. So you're wanting to be making sure you've not got any areas where a whole lot of your plot is shaded, for example, and other parts aren't. So you don't want a whole lot of trees growing down here if you can possibly avoid it. When it comes to replicating, it's then that you need to think about uncontrolled variability that exists within the sample that is here. You know, there may be some areas of the plot that are drier than others, or it may be that there are areas of the plot that are slightly higher up, because it's very unlikely you're going to get just a flat field. There may be differences within that field. So what you're going to be doing is setting up several replicates. Now, normally the minimum of number of replicates would be 3, and they must take into account the variability and reduce the effect of this experimental error. So minimum of 3 replicates, and that allows a valid statistical analysis of the results to be carried out. The more replicates that are set up, the more reliable the results. So we move on to have a wee look at the randomisation of treatments. Now, if we were in the situation that we were talking about before, where we had these 4 treatments, so we had our high concentration of fertiliser, we had a low concentration of fertiliser, and then B and C, the treatments that are in between. So there are these treatments that have been set up, the 4 treatments, and at the same time you've got these 3 replicates here. But you want to set this up in a way that you don't have some sort of orderly basis to your plots in your field, because there could be some bias there. It might be that in particular this row here has got particularly good water content in it, and yet this row here might be a little bit rockier and have a bit less water content or whatever. Or it may be that your field's on a slope, and therefore this area down here is a bit wetter than this area up here, or vice versa. So what you want to do is randomise where your plots are going to be. So you would have it maybe set up something more like this, where you've got your 3 treatments in variable parts of the field, and this can be done either by some sort of computerized generation of where you should have your plots are, or trying to allocate the plots as randomly as possible to minimize the bias that is there. Yeah, but after you've carried out this field trial, it might be you've only done it in one environment, so you might have been in sandy soil and in a kind of temperate climate. Then your conclusions that you can draw from that would be kind of a wee bit limited. So you might want to be looking at testing that new cultivar in soil that has got some kind of other high constituent in it, so less sandy soil, or does it grow well in a colder climate or whatever. So the trial would need to be carried out again in a different environmental condition. Once you finish this video, you can have a look at the article on potato blight, because we're going to do an in-class activity looking at how we can save our spuds.